घंटे क्या क्या छो इतना कर्नूल में रहो तो क्या तो खान खा छो भाई। अच्छा ठीक ठीक है। अच्छा अच्छा जल्दी आ लो।
Good evening, everyone. Welcome you all to week 6 NPTEL live session, which is on RNA splicing, export, and stability. This is Subhasmita Mahapatra from IIT Indore. I am a PhD scholar here, and today we'll be discussing few questions on RNA splicing, export, and stability. I hope my screen is visible to everyone. If my screen is not visible or if I am not audible to you people, then you can let me know by just writing down the in the chat box. Okay? I hope it's okay with everyone. Okay? If you have any doubt or any question, then you can also write in the chat box. Fine? Okay then. People are joining, so that's why I was waiting for them. But it's already 6:10, so we can start. I hope my screen is visible to everyone, right? Okay then. So let's talk about introns first, because we have been discussing about alternative splicing mechanism, and RNA splicing. This is a important part of post transcriptional modification, and we have been discussing this from 2 to 3 weeks. So I believe that you have some concepts clear about RNA splicing right now, and so I'm just briefing about today's session, what we are going to learn from today's session, that is alternative splicing. We have already talked about it, but still if you have questions then you can ask me. Introns promote transcription or gene expression. Okay, introns are those non-coding sequences present in the pre-mRNA, okay, or nascent mRNA, right? And exons are the coding sequences present in the nascent mRNA as well as in the mature mRNA. So where this transcription happens inside the nucleus, obviously we are talking about the eukaryotes, right? Because splicing mechanism only happens inside eukaryotes, right? So after transcription, this pre-mRNA or nascent mRNA, this has both coding sequences and non-coding sequences. These non-coding sequences are called introns and coding sequences are called exons, right? So these non-coding sequences are large in number, and we need to remove them before translation process, and this process is called splicing mechanism, and from which these introns have been removed out. But it has been also found that introns promote the transcription and also gene expression. How this have been happening, you will be discussing about this in few slides above. Okay, next is transcript fidelity via nonsense mediated decay. So what is nonsense mediated decay? We are going to read about it. Exon shuffling to give rise to multi-domain proteins. right? We have already talked about it, that if there are multiple exons like 1 2 3 exons, for example, there are 3 exons and they have been bounded by 3 different introns. So if I just shuffle the exons, like combine 1 with 3 or combine 1 with 2 and then combine 1 2 3, so in all the 3 cases we are getting different types of proteins, different domains of proteins. Okay, that's called exon shuffling leads to multi-domain proteins. That's why in case of eukaryotes and complex organisms we are getting a diverse range of proteins from a single gene only. Okay, so that specifically reduces the complexity of having a large number of proteins but lesser number of genes. Next is increase recombination to create more products. As you all know that recombination is a key towards different types of new evolutionary properties, new evolutionary traits. So that's why, and also in case of genetic engineering and all, recombination has given us a lot of chance to face different types of challenges, right? So here introns also been helping in increasing the recombination process to create different products. Fine. So this is all about our brief description about why genome need to have introns. What are the beneficial properties of introns? Okay, we will discuss each of these points one by one, but via different types of question mechanism. Okay, like question and answer. So here is your first question. Introns increase the transgene expression by the rate of 10 to 100 times, 5 to 10 times, 100 to 1000 times, or none. Which one is it? Please try to answer this question in 2 minutes, then we'll discuss it. Write your answer in the chat box. Others, please try to answer this question. Okay, then let's discuss about it. So here is your answer, that is 10 to 100 times. Okay, those who have answered A, it is correct. Let's talk about it more. Why genome need to have introns? We have been discussing about it just before a few minutes. So apart from that, intron mediated transcriptional regulation, how intron regulate the transcription process, these things have been covered clearly in your lecture videos. Okay, week 6 NPTEL lecture videos, these videos are clearly been explaining everything about what I am talking about. Okay, so you can go to that videos and watch it, so that it will be clear your concept about these points which I've been talking about. Understood? Here this is just a interaction session, as it call it live sessions. Here I'll just brief about the concepts and we'll discuss few questions which will help you to do your assignments. Okay, fine then. So intern-mediated transcriptional regulation, in case of budding yeast, 95% of genes are intronless, but 5% genes produce 27% of mRNA in yeast. Now you know, right? In case of budding yeast, 95% genes are intronless. That means there is no intron. But if you see, this 5% remaining genes can produce 27% of mRNA in yeast. So how come this small amount can produce a large portion of mRNA in a particular organism? Because of this introns will helping in different types of splicing mechanism, also transcriptional regulation process. Next is introns enhance transcriptional outputs. They help enhancing the transfer. What is the transcriptional output? Generally your mRNA, okay, mRNA, rRNA or tRNA. These are the transcriptional outputs. Expression of intronless transgene is 10 to 100 times lower that intron containing counterpart. That means when there is a intron, it increases your expression of your transgene by 10 to 100 times. And introns increase translational output, facilitate stability or export of mRNA. Now you got it. This also helps in translation process. Fine. So uh, when this mRNA get transferred to cytoplasm for translation process, after splicing mechanism, when there is capping and tailing also done, after that only this premature mRNA transfers to mature mRNA, and this mature mRNA then move to your cytoplasm. Okay, this cytoplasm has ribosomes and other translational machinery which helps in the translation process. Understood? Let's move to next question. UNSNRNA binds to which splice site? 3' splice site, 5' splice site, branching point or adenine residue? See, we have been talking about different types of splicing mechanisms, spliceosome and all. So what is snRNA? That means small nuclear RNA, and there is a name UNSNRNA. Why this is UN? Because this UNSNRNA basically present inside the spliceosome, and I think we have discussed about spliceosome in previous classes. So just think about it and try to answer the question. Okay, let's discuss about it. Some of you have said B, some of you have said C. Let's see. So, intron mediated transcriptional regulation, in that there are two types of transcriptional regulation process. One is splicing dependent and then one is splicing independent. Okay. So, in case of splicing dependent, as you can see, U1SN RNA, this is a small nuclear RNA which is present inside the spliceosome. So what is spliceosome? This is basically ribonucleoprotein where RNA and protein combinedly present and act together to produce multiple ribonucleoproteins. They combine together to form a spliceosome. Okay. So this ribonucleoprotein then helps in the splicing mechanism of pre-nuclear mRNAs. Understood? We have been talking about different types of introns and pre-mRNAs and all. Okay, try to remember. So UNSRNA, which binds to the 5' splice site. Okay, this binds to 5' splice site, interacts with the general transcription factor TF2H. Do you remember about TF2H? This TF2H is basically present in RNA polymerase. This is a subunit of RNA polymerase 2 in case of eukaryotes, and it helps in your helicase mechanism. Okay, that means it binds to DNA and opens it up. So binding of UNSRNA to 5' splice site helps in the transcription process to move faster. Okay, this is splicing dependent mechanism. Next one is splicing independent, where presence of enhancer or promoter element in intron. And it has been found that this enhancer or promoter, when it is present in the intron, then it helps in regulating the transcription process. Okay. So basically what happens, generally exons and introns are one after another, it is like in alternative pathway. So they are present inside a gene, and when there is a promoter or an enhancer region already present inside the intron, that helps in enhancing the whole gene expression. This mechanism is already there, so we'll be talking about it in upcoming slides. Okay, so here is your answer, that is 5 days splice site. Next is, what is the first reaction of splicing mechanism? It's been 2 to 3 weeks we have been discussing about RNA splicing. So I hope you everyone can answer this question. What is the first reaction of splicing mechanism? Electrophilic attack, nucleophilic attack, transesterification, hydrolysis. I am talking about the first reaction. Okay. Okay, let's discuss about it. So I see that most of you have said C, that is transhistorification. See, try to understand the question. Okay, here it has been asked that what is the first reaction? Cause splicing mechanism. This splicing mechanism is a combination of reaction. Okay, so this combination of reactions it happen at one after another. So here I am asking you what is the first reaction, not the prominent reaction. Like some of you have been correcting the answers again, like B. Okay, so let's talk about it more so that everyone can understand. See, this is the splicing reaction. A minute. So if you see here, this is your in exon 1 and this is your exon 2, and it is present at the 5 days end region, and this is present at the 3 days end region, and this is the intron. It is clearly visible to you in a molecular mechanism pathway, as in here you can see the phosphate group, that is PO4 minus. Okay, so now you can clearly understand what's been happening. First one is your nucleophilic attack reaction. nucleophiles. What are the nucleophiles? Nucleus means positively charged. Nucleus loving. Philic means nucleus loving. That means negatively charged molecule. What is the negatively charged molecule here? Your oxygen. So at first this nucleophilic attack happens on the 3' branch point or 3' exon splice region. You can see here that this is the 3' phosphate group, and this OH group, 2' OH from adenine residue of branch point attacks on the 3' exon point, that is your 3' phosphate group. Understood? So this is your first nucleophilic attack, followed by first transesterification. First transesterification means, if you see here, you can understand that the phosphodiester bond has been broken here and another phosphodiester bond has been made here. This AOH, that is the adenine, it has earlier it has OH groups only, but after this reaction process, you see here, you can see there is another phosphodiester bond been made along with your adenine residue. Understood? So here phosphodiester bond transfer been happened. So this is the transesterification reaction, followed by another transesterification reaction where 3' OH group attacks the 5' PO4 minus group. Here it is the arrow mark. And so after attacking, what happens? The transfer of phosphodiester bond happens, and this is the excised lariat formation, and spliced intron been combined together with a ligase enzyme, that is making another phosphodiester bond between two nucleotides to follow or complete the break point. Understood? So this is a splicing reaction overall. If anyone have any doubt regarding this splicing reaction, or if you want to repeat me once again this reaction process, then I can repeat it. Just let me know, okay? So what happens? Two successive transesterification reaction happen. Phosphodiester linkages within the pre-mRNA are broken and new ones are formed. Okay? And 2' OH group. So if someone asks you which OH group first attacks in the nucleophilic reaction, always remember it is the 2' OH group of your branching point adenine residue. Attack phosphoryl group, that is your PO4 minus group of guanine at the 5' splice site. If you remember, in last class we have talked about GT-AG rule. That means this GT, that is guanine, thymine, AG, that means adenine, guanine. These four residues are conserved in case of nuclear pre-mRNA, that in the exon intron boundary. If you see this GT and AG in the exon intron boundary, then that means it is a nuclear pre-mRNA and it is conserved. Understood? So this is generally happens in case guanine residue is present. So and in later on, this 3' OH group of the 5' exon attacks the phosphoryl group at the 3' splice site, and it has been removed as a lariat intron, and spliced introns been ligated towards each other with a phosphodiester bond. So this is the whole splicing mechanism. Understood? So first reaction is your nucleophilic attack reaction. I think it is clear to everyone. Let me know if anyone have any doubt. Okay, so here is your next question. Which class of intron are more prominent in eukaryotes? Group 1, Group 2, nuclear pre-mRNA, or all? We have been discussing about introns, so try to answer this question. Which class of introns are more prominent in case of eukaryotes? Okay, then let's discuss about it. I see B or C. Some of you have said B, some of you have said C. Let's see. So this is the class of introns like nuclear pre-mRNA, group 2 introns and group 1 introns. So in case of abundance, if you see, nuclear pre-mRNA is very common, used for most eukaryotic genes. Group 2 introns are rare. Some eukaryotic genes from organelles and prokaryotes have group 2 introns. Next is your group 1 intron, that is rare. Nuclear rRNA in some eukaryotes, organelle genes and few prokaryotic genes. Okay. Two mechanism. Two transesterification reaction. Branching site is A, that is adenine. So which group of adenine at first act on the process? That is your 2' OH group. Understood? And group 2 intron, it is same as pre-mRNA. It has major and minor spliceosome. RNA enzyme encoded by intron, that is ribosome. So if you see in case of group 2 intron, ribozyme is needed. But in case of nuclear pre-mRNA, spliceosomes are needed. And they are generally made of two types of spliceosomes, major and minor. We will be talking about in few minutes. Okay, next is group 1 inter. The mechanism is 2 transesterification reaction. Branching site is guanine. So now you understand the difference between group 2 intron splicing mechanism and group 1 intron. In case of group 1 intron, the branching site is guanine. In case of group 2, it is adenine. Okay. Next, catalytic machinery is same as the group 2 intron. Fine. So here is your answer, that is nuclear pre-mRNA. Next is splice ladder RNA is used in which splicing reaction? Cis splicing reaction or trans splicing reaction? Which one is it? Try to answer this question. Let's discuss about it. I see that some of you have answered B. Others, please try to answer. I see some people only answering the questions. If you don't know the answer, you can guess it. It's okay. We'll be discussing it, right? So, let's talk about it. Splice ladder RNA is used in which splicing reaction? Cis splicing or trans splicing? So, for your information, I would like to tell you that cis splicing. First, we need to understand the basic concept of cis splicing and trans splicing. So, in case of cis splicing, what happens? Your own machinery, that means the 2 days OH group or 3 days OH group of different exon regions, that is 3 days splice site or 5 days splice site. If you see here, that everything is inside from the gene only. That means, for example, if this splicing reaction been happening inside the nucleus, then if you see here that not much outside agents been used in this process. Okay, and only the 2 days OH group of this adenine residue and this 3 days OH group of this exon sites have been used for doing the transesterification reaction, which led to the Lariat formation of intron and spliced combination of this exon regions. Okay, so if you see, this has been happening using the inside mechanism only. That's why it is called your cis splicing. So what is the trans splicing means? Trans splicing means when some part of your intron, that is 5 days splice site or 3 days splice site, is missing, and this particular exon required to another exon, like see here. In case of this pink one, if you see, this is a splice ladder. Splice ladder RNA in transplicing is a mechanism of gene expression, and it is a form of RNA processing in which a splice ladder is joined to 5 days ladderless pre-mRNA to form the mature 5 days end of mRNA. That means when your 5 days end is missing or ladderless, then this splice ladder can be used to complete the process. And this process adds a conserved sequence to the 5 days end of mRNA, the splice ladder ranging from 10 to 50 nucleotides, and bring a 5 days cap to the mRNA. Fine. So let's talk about it. This is cis splicing. Here if you see, this at 2 days OH group of adenine residue been attacking the 5 days site, and the process continues, right? But if you see here in the trans splicing, there is another splice ladder, that is your pink color one is showing here. Fine. That's why this adenine residue, it has 2 days OH group. So this 2 days OH group is now attacking the 5 days region. 5 days exon site of this another different splicing ladder, that is splice ladder you can call it. Now see here, after this bond, this phosphodiester bond is been broken. This, if you see this PY, you will understand this phosphodiester bond is been broken. Okay, that means this part is broken, and so this exon part, where this will go, this will move to your 3 days exon site, and now using the ligation process, this is your new exon 1 or another new exon, which will lead to when it gets translated, it will produce another different types of protein, and this is your intron which have been removed. Understood? Let me repeat it again. In case of trans splicing, there is one ladder sequence present, which is shown in pink color or red color here, and there is some part of it that is required for the splicing mechanism. Here this adenine 2' OH group attacks on the 5' site of the new exon, and this new exon then phosphodiester bond has been broken, and this new exon then move to your 3' exon site. As you see here, this has been form a phosphodiester bond with your adenine residue. That's why this will remain here, and this exon will come to the black exon that is present at the 3' site, and they both will combine to produce another mature mRNA, which when get translated it produce different types of proteins. Now you know a new mechanism of getting different protein diversity using a single gene only. Understood? This is cis splicing and this is trans splicing, and this has been happening inside your trypanosome, C. elegans, flatworms, hydra, and primitive chordates. Understood? So this is all about trans splicing. Next, introns help in exon shuffling and recombination. Is it true or false? Try to answer this. Okay, as let's talk about it. So it is true. 1 minute. Exon shuffling, it creates new combination of exons by intronic recombination. We have been talking about this alternative splicing from 2 weeks, so now I think you understand about why the importance of alternative splicing. So exon shuffling was vital in creation of multi-domain genes in metazoans. Introns increase the recombination rate in coding region of genes. Okay, so in exon shuffling means when it combines multiple exons to produce new mRNAs. An exon is duplicated within the same gene. Now you see, so this is exon A, this is exon B. So this exon A is here, but B is here, but this exon B is duplicated. Duplicated means one it in there has been two copies. So because of these two copies of a particular B chain and a particular A chain is there already, so this will lead to a protein, for example X, and after combining new exon is inserted into the existing gene. So how come this new exon came into picture? We have been just discussing about this transsplicing. So transsplicing is the mechanism where this new exon can come between two uh, old exons. So here this A exon is there, B exon is there, but in between them there is another exon called C. So now this will lead to a protein Y. Got it? So how come this combination of this A and B exons can produce two types of proteins, that is X and Y. So like this we can combine different shuffle the exons to produce different multi-domain proteins in case of different organisms. Okay, here is the next question. Introns at which region enhance gene expression? Now we are talking about the importance of introns in transcription regulation and helping in gene expression. So try to answer this question. 5' site, 3' site, promoter region or terminator region. I think it is a simple question. Everyone try to answer this question. Okay, let's discuss about it. As we have been talking about this intron and gene expression, so which region enhance the gene expression? If you just use your matlab, simple logic, then it must be your promoter region. Okay. So when this promoter region has introns, then it enhances the gene expression of the exons also. That is the coding sequence. This is the process of intron mediated enhancement. If you see here, this intron this is called a looping process. Okay, this is called looping process. Here this intron near the 5 days end of gene recruit transcription machinery to the promoter, thus to initiate transcriptional initiation. So here introns are present in the 5 days end of the gene, and it recruited transcription machinery to the promoter. That means it just loops it out to move towards the promoter so that the transcription initiation can happen, and this will enhance the gene expression. This is the promoter, terminator, and 5-day sequence and 3-day sequence. This is the intron. Because of this looping, this has come closer. Okay, fine then. Let's move to next question. Here few short forms are there. That is, what is the function of NMD pathway? Promote transcription, promote translation, degrade the prematured translation products, or none. Okay, let's discuss about it. I got only one answer, that is C. Degrade PTCs. At first, talk about the NMD pathway. What is NMD pathway? This is your nonsense mediated decay pathway. What is nonsense mediated decay? So, earlier in the last class, we have been talking about truncated mRNAs. If you just remember this, there have been truncated mRNAs and truncated proteins also. When in the splicing mechanism, if a stop codon appear, okay, if the stop codon appear inside your codon region, that leads to your stop of this translation process in the middle of this mechanism, okay, and that leads to a truncated protein or truncated mRNA. Got it? So in NMD pathway, that is nonsense mediated decay pathway, it degrades the mRNA containing premature translational termination codons. Understood? So, what is the function of this pathway? To degrade the mRNA which contain the premature translation termination codons. It is more efficient if the stop codon is present upstream of the exon-exon junction. Monitoring of mRNA with PTCs is done in the pioneering round of translation. So, uh, as we have already discussed about it, I am just trying to brief you about this mechanism, that when there is a particular stop codon is present, or sometimes what happens, there is when there is a particular GC rich region, that is guanine and cytosine rich region, that produces a hairpin region, and because of that the transcription stops there, and that leads to your stop truncated formation of your mRNA. That is one reason. Second reason is when there is any stop codon rare, like your premature translational termination codon. Which are the stop codons? UAA, UAG, or UZA. When these three codons are present anywhere in the exon region, if you see here, this is exon junction or exon prior the exon or inside the coding regions, so that may lead to production of your truncated protein or a disabled protein. So in that case, this NMT pathway degrades the mRNA containing the premature translational codon, that is PTCs. Understood? Fine, I think it is clear with everyone now. Okay, then let's move to next question. So what is the function of transplicing? We have been discussing about transplicing few minutes before. So now tell me, what is the function of transplicing? Regulation of gene expression, 5 days gap provided by splicing ladders, helps in separation of cistrons, all. Everyone try to answer this question. I think we have been discussing about this in few half an hour, so you can answer this question. Okay. So I see that most of you have said C, D, that is all. Yes, it is correct because splicing helps in gene expression. Using the splicing ladder, this 5-day escape have been done and helps in separation of cistrons. So now let's talk about what is the difference between cis splicing and trans splicing. So in case of cis splicing, this is this U1 snRNA, U2 snRNA is already there. When cis splicing involves two exons are joined by eliminating the intron, while transplicing involves using a leader segment from one source and joining to the 5-day end of another pre-mRNA which lacks the 5-day ladder sequence. So if you see here, in case of transplicing, this particular mRNA or it doesn't have this 5-day site. Okay, this 5-day leader segment is absent here. So in that case, a foreign or new snRNA can help the other pre-mRNA in the splicing mechanism. In cis splicing, the UN snRNP, that is UN small ribonucleoprotein, base pairs with the 5-day splice site, and U2 snRNP base pairs with the branch point near the 3-day splice site. This is your 3-day splice site and this is your 5-day splice site, as you know. The intron is excised and the two exons are spliced together. So if there is UN and U2 snRNP is already present, that means we are talking about your spliceosome mechanism, spliceosome mediated splicing. Okay, spliceosome mediated splicing. But in case of transplicing, there is no 5-day splice site on the pre-mRNA of UN snRNP. If you see here, there is no 5-day splice site. Instead, the 5-day splice site is provided by the donor SL snRNP. This is your donor SL snRNP, which interact with the U2 snRNP at the 3-day splice site, and the SL exon is spliced to the exon of pre-mRNA. Splice 2 means ligated to the this exon, that is exon 1 will combine with this is your exon 2. The region between 5-day gap and transpliced site is called outron. So now you know a new word from transplicing, that is outron. So the region between the 5-day gap and the transpliced site. Okay, so this is the region which is called outron. Fine. Now you know the difference between transplicing and cis splicing. Let's talk about few more mechanisms. So now we have been talking about spliceosome mediated splicing mechanism. So there are two types of spliceosome generally, that is your major spliceosome and minor spliceosome. So now tell me, major spliceosome is made up of which type of introns? U12 type of introns or U2 type of introns? What is it? Okay, let's see. Okay, this major spliceosome is made up of U2 type of introns and minor spliceosome is made up of U12 type of intron. There is nothing to understand in this case. This is basically the types of introns present in which type of spliceosome. Okay, so this basically this is your answer. And this is the snRNPs present in the spliceosome. snRNPs or SNRPs, they are small nuclear ribonucleoproteins, and there are different types of snRNPs like U1, U2, U6 and U6, or you can call it as 12S. And one thing is quite you know interesting, that the size of spliceosomes sometimes similar to the size of ribosomes, because ribosomes are made up of rRNAs, right? And they produce help in the translation machinery, and they have this in case of eukaryote ribosomes, there is this 80s and 70s ribosomes are present in the prokaryotes like this way. Here also 12s, that is the sedimentation rate, right? So here also 12s snRNP is present, 17s, 13s, 12 20s and 25s is there. So as the sedimentation rate increases, if you see in case of 13s, you have two snRNAs, that is U6 and U4 combined together, and here U5 is there, and here three snRNAs combined together, U6, U4 and U5, and this is the structure of snRNA already present. Okay, and these are the subunits written here. You can look into it in detail, but I would say that just try to remember the sedimentation coefficient with the subunits, like 13 has as U6 and U4, 17 has as U2, and 12 has as U2 U1, and 25 has both three, all three, that is U6, U4 and U5. Understood? Fine then. Let's move to next question. Which spliceosome has equal size as ribosomes? Major spliceosome or minor spliceosome? This is very you can say uh, you know very easy to guess this question, but I would like to everyone answer this question. Just think about it a little bit and you can answer. Okay, so I see most of you have answered A, and it is correct. Very good. Major spliceosome. So what is this major spliceosome? It comprises of 5 snRNAs and more than 170 proteins. Spliceosome size is similar to the size of a ribosome. And RNA splicing is ATP driven. That means ATP is required for the splicing mechanism. RNA participates in splicing reaction. Also, these snRNAs are 100 to 300 nucleotides long, complexed with proteins that leads to production of your small nuclear ribonucleoproteins, that is snRNPs. Okay, so and these multiple snRNPs combine to produce a spliceosome. Like this snRNP, for example, it is 10 snRNPs combined to produce one single spliceosome. Okay. Here is the last question of today's session, that is snRNAs U1, U2, U4 and U5. Earlier we have been talking about different types of snRNAs according to their sedimentation rate, right? So this 4 snRNAs U1, U2, U4 and U5 combine to form a snRNP or SM core complex or SMN complex or spliceosome. Which one is it? Okay, so let's discuss about it. I see a diverse range of answers coming like A, C, and D. Let's talk about it. SM proteins, snRNPs, and the spliceosome and messenger RNA splicing. snRNAs like U1, U2, U4, and U5 were found to tightly bound to several small proteins known as the SM core protein or simply SM protein. Okay, then these snRNAs are complexed with SM core protein and with other proteins to form particles in the cell's nucleus called SNRPs or snRNPs. Understood? How come this snRNPs been formed? Okay, at first they bind to the small proteins to form a SM protein or SM core protein. Then these SM core proteins combine to produce snRNPs. Got it? By the mid of 1980s, it is becoming clear that these snRNPs help form a large complex called spliceosome. Now these multiple snRNPs combine to produce a spliceosome. Okay, around pre-mRNA, excising portion of the pre-mRNA called introns and splicing the coding portion, that is exons, together. After several modifications, the spliced pre-mRNA get transformed to messenger mRNA, and which is then exported to the nucleus and translated into protein by ribosome. So understand that and don't be confused. After all this splicing mechanism, when this mature mRNA become produced, that will be transferred to the cytoplasm, not the premature mRNA transferred to the cytoplasm. Okay. Next is assembly of snRNPs in the SMN complex. What is it? So this snRNAs and like U1, U2, U4 and U5 and less abundant U11, U12 and all interact with the SMN, that is survival motor neural protein, and gemins to produce the SMN complex, and this SMN complex then guides towards the production of SNR protein 1. You can read it and you can understand it clearly just watching the lecture videos, okay, of week 6. I am just trying to brief about the lectures, not I cannot describe each and every detail of the SMN complex or everything, as the because of this time limitation and the particular question answer session is there. Okay, and if you see here, this is the pictorial representation of production of SMN complex and snRNPs. See here, this is the SM protein 6S complex, then 8S complex, next lead to this SMN complex. You can read it and understand it. So today we have been discussing about the different types of splicing mechanism and different types of proteins being used. Okay, earlier we talked about alternative splicing, spliceosome, and different types of introns and exons and like that. But here we discussed about different proteins involved in the splicing mechanism. Okay, so try to remember these mechanisms. This will help you in understanding the RNA biology more and more. Okay, thank you guys for joining. uh, I think we can end today's session here. We'll have the next meeting on next Monday.